To our next speaker, Dr. Abdul Mannan Baig. Dr. Baig is a medical doctor and a scientist from Pakistan. He has served as a physician in internal medicine at the University Hospital in Pakistan. He earned his PhD from the University of Sunderland, and after the outbreak of COVID 19 in early 2020, he has published over 25 papers on COVID 19 and SARS CoV 2. His papers on COVID 19 and SARS CoV 2 focused on clinical presentation, treatment modalities, and targeting vital targets in SARS CoV 2 caught attention even in the international news outlets and researchers worldwide. Recently, he has published on neurological defects in the long COVID in Nature as well. Dr. Baig is a recipient of the National Research Productivity Award from Pakistan Council of Science and Technology and the Award of Excellence in Research from Aga Khan University. So Dr. Baig is going to talk about NeuroCOVID, actions needed for prevention and treatment of neural damage. So let's welcome Dr. Baig.
Okay, so this slide shows you the progression of a disease which was initially thought to be a respiratory disease, and it shows that how you acquire the infection to how it goes to the brain. And in the slide, okay, you can see from the region of the nose.
So yes, so whilst we're waiting on Dr. Mannan Baig, I'll look and see if there are any quick points. We've got a few points here, shining bright light on the whole thing. Yes, thank you, Andy. And we've got Equilibrium. Thank you as well for sharing your thoughts with us. Mitch, thank you very much for being with us. We appreciate your presence. And is that better now, Manan? Can you go again?
Yeah, I can hear you. Can you hear me now?
Excellent. I'm hearing you fine. So you go ahead and I'll move your slides.
Okay, thank you very much. So this slide shows you that in the phase of acute COVID, one can have neurological symptoms as shown in diagram B. And it could be attack of the virus on A. Once the virus is inside the cell, it can cause development of certain deposits, okay, that you can see in the diagram B3, Philip. They include deposition of plaques like beta amyloid and even tau protein. So there is a question that is asked that if it goes beyond 3 months or years, okay, is it possible to diagnose that the neuroinflammation is still going on? On the spot B4, you can see I've shown some markers, okay. What I was saying is that the CSF can have some elevated biomarkers as shown at a diagram spotted as B4. The question is that is the direct viral injury only explaining the neurological symptoms in long COVID? So it might be the to the brain, okay, caused by thromboembolic phenomena. If Philip could spot it on C, you can see that embolic phenomena and thrombotic phenomena can also give rise to hypoxia. Hypoxia can cause a neuronal damage. So the question is that with NeuroCOVID, what the person is exhibiting in form of sign and symptoms that you can see now. Philip is showing you that there is a brain fog. I personally don't like this term because this shows you the key players, okay? Persistent neuroinflammation, presence of SARS-CoV-2 and its associated protein. My earlier speakers, my peers, actually spotted the role of S protein and N protein of SARS-CoV-2. You can, at the upper right-hand corner, see the vascular event ongoing that you just heard can cause ischemia, hypoxia, and cause neurological deficits. So if you could move to the next slide, I could show you how the damages are taking place. Now, if you see, okay, the infection goes through cribriform plate and it can infect the olfactory bulb as shown in the upper right corner. But there are places, okay, where the virus hideouts. You can see the and reside and can come back and then reinfect the newer cells. So these hideouts are important because the virus can emerge from here and cause cause a reinfection of the naive cells in the body. The reason many long COVID patients are having problem with sometime feeling better, at other time feeling worse, like that. Next slide, please. So here is shown that what sign and symptoms could occur in NeuroCovid. You can see them being listed from number 1 to number 12. Okay.
Yes.
So this slide actually shows you that what type of sign and symptoms the patient can suffer in acute COVID and carry it over to long COVID. And in the next slide, if you move on, you would see the evidence. Okay, many people ask that where is the evidence? Show that whether it goes inside the neuron and infects it. Okay, so at the regions of the star, you can see below in the black and white diagram, the inset is showing you the presence of SARS-CoV-2, not the RNA of it, the entire virus inside a neuron. So this paper was published, and it came out in for COVID-19. But then just imagine that the same type of infection the person can be carrying into long COVID. Next slide, please. No, this slide, okay, when I just made it, okay, I saw my earlier speaker went into quite a detail of it. You can see not only the virus itself, but the spike protein itself can cause formation of fibrils, which then subsequently become the beta amyloid and tau protein. At the top where the mouse is moving, okay, you can see spike protein can blend up with the amyloid fibril, and in the presence of a neutrophil elastase, which is a very crucial player, okay, can make super fibers, or these super fibrils, okay, get deposited in the neurons, and they assume the form of beta amyloid and tau protein. So once these substances get accumulated, just imagine how quick the neuron would age. So a person in 30s and 40s would be like having neurons, okay, which are laden with these deposits, okay, which are reminiscent of Alzheimer's disease. You heard Stephanie saying Parkinsonism, and with Lewy body and stuff, okay, they would resemble many other degenerative diseases. The $1,000,000 question: what this virus sets up into the neurons? Okay, it sets up apoptosis, as you can see in the diagram in the below left, where the a red arrow is coming out of the skull of the human, and it also causes the loss of brain mass, okay, which was shown for many patients with COVID-19. Now, the next diagram will talk about the role of glia and other stuff. But before that, I want you to see this, that there are several molecules, okay, which can target the pathways and the virus and the S protein as well. You can see the name of the molecules highlighted in blue and their chemical structure. They are very well known inhibitors of cytokines that are listed in green. There are certain drugs like piperine, okay, and hesperidin, rutin, which with the sign of inhibition are shown that what process they can inhibit. So I was just wondering that instead of these drugs alone, okay, if there would be a formulation which have these components in some nutritionally allowed quantities, remarkable effects could be observed. A newer player into the game is known as immunomodulatory drugs, which is shown on the top right with the name of IMiDs. Okay, one example of the drug is polymedomide and thalidomide family. It also has got a lenalidomide family. These are neuroprotective as they inhibit the alpha TNF, but then I came across very few of them from the literature that are as neuroprotective as the nutraceuticals that you can see on the slide. Like hesperidin inhibits the SARS-CoV-2 main protease as well as the spike protein. Rutin, you can see, is anti-apoptotic. It's doing the trick here by inhibiting the microglia-induced apoptosis. On the extreme right, you can see two major players, okay, the piperine and Vicalin, okay, doing the inhibition of the cytokine. So if we manage to not only target the virus, but also inhibit the inflammatory process, that could be a success in treating long COVID with neurological symptoms. Next slide, please. Now, dysautonomia is thought to be autonomic nervous system disorder, but then you see the surname says nervous system disorder, so it also comes as a subclause of NeuroCovid. many patients, okay, countless of them, okay, are in long COVID are suffering autonomic disturbances. You can ask me a question, okay, how does the autonomic symptoms arise in patients with long COVID? The next slide will show you where actually the virus can target. Just look at the mid-sagittal section of the brain. You can see vasomotor center there written. Vasomotor center is an area which controls the autonomic nervous system. There are two very high players, okay, in the hypothalamus. Highest control showed as green and yellow. And just imagine that if these neurons get infected with SARS-CoV-2 or their spike protein, they are going to just like bring an earthquake to the autonomic nervous system, regulation of autonomic functions. The reason patients suffer not only tachycardia when they stand up from a lying position with POTS, and they have got anatomical and physiological instability with their management of their blood pressure, number one, their heart rate, their even venodilatations. All of them are kept in check by the autonomic nervous system, which seems to be a very clear and evident target of SARS-CoV-2 in long COVID. Next slide, please. Now, this one is showing you that don't mix this amyloid with the cerebral amyloid that I was explaining, but in the periphery, okay, with chronic inflammation, there could be formation of amyloid depositions, which if occlude a coronary artery can lead to a heart attack. But our topic today, NeuroCovid, if you go to the top, you see it can even occlude a cerebral blood vessel leading to hypoxia. Now, don't mix up this one with stroke. This one actually partially blocks the blood vessel, so the blood flow to the brain gets limited, and then the neurons can suffer hypoxic damage that you heard Stephanie and other speakers were stressing that it can cause a neuronal atrophy and deposition of beta amyloid and tau protein. Next slide, please. Now, the treatment approaches. So what can be done? I mean, if you ask me a magic pill, okay, that would be a pill that has got anti-inflammatory effects, neuroprotective effect, could be antiviral at the same time, very preferably a nutraceutical, if not stem cell therapy, okay, which I'll talk in the next slide to show you. If these drugs and even the stem cell therapy could go into a clinical trial to see that whether it could improve the symptoms with NeuroCovid or not, it would be some practical thing to do. Next slide will show you that how the long COVID or NeuroCovid symptoms can be addressed. Number one, you should be targeting the viral loads. Number one. Number two, you should be targeting the re-entry mechanism, which is so very well known by the virus when it comes out from the hideouts, and of course the mother of them all, the viral replication. So how it could be done? Now on Twitter and several other social media channels, okay, very commonly you see posts, okay, especially when people ask that how viral loads can be eliminated. Simple cleansing of the nose, okay, no need to use hypertonic salines. Simple inhalations, they can do the trick, okay. If you do it daily, it reduces the viral load. I want the audience to understand one thing, that viral loads are not constant. Once you get an infection and it's not going out after 3 months or a year, that means your body in sites of viral loads, be it colon or gut or nose or sinuses, it's continuously like the virus is coming out of the cells after infecting them in a very large quantity. So the viral loads may be building up and you might not be knowing it. You only suffer symptoms and you think that it's some cytokine thing. The cytokine is driven itself by reappearance of the virus which comes out of the hideouts. Re-entry mechanism is my targeting it is my favorite. I'll tell you why, because I've been experimenting with drugs and some in vitro results that if you block the S protein of and flip, if you can just click, okay, those molecules would appear. You just press the space bar. Yeah, you see, it's an animation slide. Now you see the drugs coming and blocking the S protein of the SARS-CoV-2. So if they block the S protein, it cannot sit over the ACE2 receptor. It's an inconvenient word, but I call it the castration of SARS-CoV-2 because it would not be able to do what it actually is destined to do. So it can forget about sitting over that ACE2 receptor, so it's as good as a dust particle. So there are drugs which can actually block this S protein of the SARS-CoV-2. You heard with you saw in the previous slide with hesperidin and piperin and rutin. They all successfully sit over this S protein and block it. If you see the third one on the extreme right hand side, you can see two nutraceuticals, okay, shown to block the main protease of SARS-CoV-2 at a site which is shown here. If you can click the space bar again, Philip, it would appear here at the socket. See, these two drugs, okay, have shown consistently to come bind at this site, okay, which has the inhibition site of the main protease of SARS-CoV-2. Next slide, please. So now, neural stem cell therapy, okay, seems to be very attractive, and many people with long COVID, okay, keep on asking the question, do we stand a chance with this? Now, there is something which I want you to know. If you could click the space bar, there is a problem with this therapy, and that problem would appear in the next slide. If you move on, Philip, thank you. You see, on the left-hand side, you can see areas where the neuronal loss are, if they are there, okay, are very accessible to the hands of the transplant surgeon. And very usually it could come out as an uncomplicated surgery if the region is like very handy to approach. But if you see on the right-hand side of the slide, in a mid-sagittal section, which shows the presence of the neuronal damage or replacement that has to be done in hypothalamus or thalamus or the lowermost black circle, the medulla oblongata. These are very critical location. And let me tell you something very candidly, that the operative procedure to attempt to transplant cells here itself could cost a life, because you can't reach these places so very easily. It's very easy to say that you can see in the mid-sagittal section. Just imagine how a surgeon would approach these areas. Not possible. So if you click the next slide, okay, you will see that this is a critical location, and post-operative complications are there. So question is that can medicines and some regenerative, what you call as growth factors, can do that? Next slide, please. So you see, I'm not saying it out of clouds, okay? There are evidence-based literature, and you can see them from top to bottom, that the compounds vacillin and hesperidin and quercetin, they have been known for stimulating neuronal stem cells, and they actually touch those critical pathways, okay, which can cause differentiation of cells into neural stem cells. So my thing is that in cases, okay, as you saw in the previous slide, when the transplant surgeon is actually thinking of attempting those areas where even going there, okay, the operative procedure could kill, these therapies, okay, would be more the mainstay of the neuronal regeneration in critical location type of transplant attempts, because then you won't actually wait for the post-operative complications to occur, because those are given in form of drugs. So if it's surface, maybe stem cell can stand a chance, but if they are deep inside the brain, forget about the procedure. I mean, like excuse me, some of them say that injecting the stem cell into the CSF can do the trick. Not at all possible, because it can go to other places and grow there and result in a dysgerminoma or a germ cell tumor, okay? So transplanting at the accurate site is needed. If not, the next slide, please. These medicines, okay, for which I've shown you evidence-based papers here, one of them in scientific report Nature, okay, clearly indicate the role of curcumin and other molecules which can cause a neuronal regeneration. As you heard me earlier, okay, my thought is that if these chemicals and molecules and nutraceuticals could be given in a proper combination, they could serve much better than the agents alone. So next slide, please. These are the molecules, and you just see where they get published. I mean, like, it's not I mean, like just a proposal or something, okay? This is a Springer Link journal where they showed molecules which are drawn and with their name, okay, to be anti-inflammatory, neuroprotective in their action. There are multipotential compounds shown on the right-hand side. You heard me on them on previous slides, okay, where their anti-inflammatory and antioxidant properties were discussed. Some of them are brilliantly neuroprotective. The reason these would stand a new chance in addressing the neurological deficits in long COVID patients. Next slide, please. Brings us to the point: who is going to do it? I mean, like, from where to get an advice? I mean, online telemedicine is going on nowadays for COVID. Even for long COVID, okay, there are institutes where you can consult for your neurological symptoms, as shown on the left hand side. Always brilliant to ask patient, okay, in a consultation, that what are the current drugs you are upon. Right hand side of the slide, please. And if these two pictures are known, I mean, neurological symptoms can give an idea to a consultant that where exactly the focus is. And if the current drugs he or she is taking is addressing viral loads, re-entry mechanism, and viral replication, very good. But if they are not targeting like any of these or all of these, then I say that there is a chance that neurological symptoms would reappear, or there is a chance of a relapse. The question: how would we know that which patient with long COVID needs what therapy? You just see at the arrow pointing below, individualized therapy. That means not every person would need the same amount of anti-inflammatory and neuroprotective drugs or a neuronal stem cell therapy. You have to individualize the patient according to his or her needs. So, but then at the end of the day, the surname of the treatment is anti-inflammatory. It should be neuroprotective. It should have a antiviral component, and if accessible to surgical hands, okay, why not even a neural cell stem therapy? So with this thought, there was a proposal, I mean, like, to how to address these things. Okay, what should the patient prepare? What should be I telling to my consultant when I visit him? Next slide, please, the last one. This one tells you that, first of all, your online telemedicine consultant should be the one which by reputation knows. If he knows, okay, book a consultation, get a Zoom session or a Skype or a team, whatever you like, the arrows which are going below. So treatment options, okay, discuss with them, and almost always request that he should brief your local consultant, who may be sometime reluctant, okay, to talk to a doctor from online telemedicine, but then just ask him that like he has got some thoughts, okay, why not just listen to them? To the patient, I would say keep it handy. Number one, your current lab results. You see in the yellow, your current lab results should be with you. Your current drugs that you are taking should be with you. The list of them. You should not be searching while having a consultation. Get them prepared, and be very prepared to ask question. I mean, just the one below it, yeah. And don't hesitate. I mean, like, even if you want to ask that, I've got dysautonomia, is the virus in the hypothalamus or medulla oblongata, or is causing demyelination of my nerves? What chance do I stand, okay, with the multivitamins, nutraceuticals, or even multivitamin injections with the myelin sheath regenerate or not? Can my neuron come back or not? Like, feel free. I mean, like, this is your time. If you don't know what you are going for, it's very, I mean, consultation into clouds. So the patient should be ready with few things. Your online telemedicine consultant should talk to your consultant in real life, okay, write him a letter or what. You can ask me a question, okay, like, how would a online telemedicine guy would examine doing a NeuroCOVID telemedicine session? You see 3 slides on the extreme right hand side, please. We can test cranial nerves. We can check for various signs like rigidity, tremor. You can save the patient to perform certain task, okay, which gives clue towards the neurological symptoms. So it's not totally impossible to have a neurological examination online. But if you go and your consultant knows, okay, what you should do, okay, in order to give him clue that which part of your nervous system is possibly affected, I think it would be a brilliant thing to do. At the bottom, you can see COVID-19 written with a link, This is one of those 75 sites that I know that perform consultation for COVID-19, acute phase of COVID-19. But we need to follow them, okay, and develop sites like these, okay, where telemedicine for long COVID should also be available. I mean, like, I mostly do it on Twitter, okay, but then there should be many country-based, globe-wide online sessions for in order to do this. Thank you very much.
Thank you, Dr. Baig. That was great. Um, so I got a couple of questions, and then I'll let you, Jim, um, ask you some few more questions. So, Dr. Baig, I just want to know, you have highlighted quite a number of nutraceuticals in your slides, and that's quite interesting because my lab also significantly focused on those nutraceuticals, and I have clearly shown that they have potent anti-inflammatory and even anti-cancer activity in various lung diseases, apart from lung cancer, asthma, and COPD. So,
Okay.
what do you think? What are the pathways you are looking here that in these neurological disorders, these nutraceuticals are targeting?
Now, that's the beauty of an alkaloid or a neuroceutical particle, that when you actually develop it from scratches, okay, you know a single pathway or two pathways that a drug could target. These compounds for centuries are known for their effects in infections, especially viral infections. Some of them were even used as Chinese medicine, okay, for influenza and other viruses, after which we noticed Parkinsonism in 1918. So you name it. I mean, like, with the compounds that you saw, they are antioxidant. At the same time, they are neuroprotective. Over 53 papers on them.
Yeah.
And I keep downloading those PDF, okay, and to just like reconfirm what else it could do, what else could do. But the charm is in their combination. I mean, that's one thing I would like to emphasize, please.
So, is there any particular molecular or cellular target you think it's a common among all?
All of them, okay. If you are talking about neurocovid, the answer would be different. If you're talking about long covid, okay, then the pathways would actually disseminate, okay. I mean, like, so
Right.
if you say or neurological or neurocovid, they are anti-apoptotic. They prevent the development of amyloid. Some of them have been reported to clear the amyloid, okay, and tau protein. So I think that for neurocovid, that's the answer. For long
Right.
COVID, the pathways are like, you won't believe. Okay, I know guys, okay, who have got more than 250 pathways, okay, on a single wall, okay, with these molecules acting.
Yeah. So, in continuation to the same question, do you think the brain of the long COVID patients would exhibit amyloid, tau, or any other deposits?
Now, this is a question that I will answer now, Kamal, and we will come back to this, okay, via either email or a similar talk in 23 or 24. The
Yeah.
answer is yes. Okay, yes in caps. Ask me why I am so sure about it. Okay, I'll tell you why. See, if a person had acute COVID, okay, and then after a year he's having similar symptoms, you tell me what actually he's carrying? A malaria? I mean, like, I don't
Mm.
understand. The same virus is there for some reason or the other. You can't detect it, or it has shedded proteins which are there, which are causing the problem. So the
Yeah.
question is that in acute COVID, you have found beta amyloid, tau protein, okay, and you name it, okay. So if it's a low grade process, you will see. I wish that my patients with long COVID live enough and get treated, never get autopsied. But the few cases that we will have, okay, in 2030, 2022 and 24, Kamal, we will have a session, okay, in which I will tell you that in 2022 I told you. Okay.
Sure. And what do you think with the brain fog? You are
Hmm.
the term which you don't like. What would be
Okay.
the basis at a molecular level for that condition, like what you call brain fog?
Okay, Kamal, I'll put an example so that you and the audience would understand it very quickly. Okay, so if I ask you, okay, there are two goats, okay, hunted by a lion, okay, and the lion actually would do what to them? Okay, so how much time would you take to say that he would catch one of them and the other would run away? Okay,
Yeah.
believe me, even this simple question, sometimes the guys would actually take time to compute to understand that what he's asking. this is brain fog. Okay, they look at a zebra crossing on the street. Okay, it looks like a tunnel vision. I mean, like, should I walk or should I stop? I mean, like, memory recalls. Okay, cognition problems on the top of them, 7 to 8 of them. Where is the fog part of it? Okay, I don't see fog. You see in a rainy weather, So this term came, it grabbed. Okay, and it's there, but it should not be misunderstood. All the brain fog complaining patients have got severe cognitive dysfunctions.
Sure. Thank you. Thanks, Dr. Baig. I'll hand over to actually Yochim. Yochim, you are on mute.
Hi, Manan. Yes, after we have spent several weeks and I don't know how many hundreds of papers we have exchanged on all this, let me emphasize one thing maybe that was not really well highlighted. We're looking at these fibrils forming, especially from the Swedish study, where you could see that the spike protein dropped in serum and watched under an electron microscope would then dissolve itself into amyloid beta fibrils. And especially if you add neutrophil elastase to that, they would become cleaved and become shorter and much more toxic. And my suspicion would be that similarly the question, will these fibers then persist, and will they go and form thrombosis, and can they form amyloid or other aggregates in the brain at the same time? Are we looking at the same main source for these two very prevalent conditions in long COVID?
Yeah. It was on one of my slides at a 12 o'clock position, okay, where I showed spike protein dissolving in amyloid fibrils and forming superfibrils. See, when I say superfibril, okay, just try to understand it, that this is something the body has never seen. $1 million question, can the body dissolve it? Okay, $1 billion answer, it cannot. Why? Because it has never seen that. So then comes the point what to do with it. Okay, you deposit it, and then it comes into traces. Okay, whenever you do the autopsy, that actually is the fate. If it dissolves in a blood vessel, what would you do? You can't remove it. I mean, like, because you have never seen it. So if you have got a product, okay, for which the body is very new, okay, to it, okay, from where it would bring the enzyme to dissolve it. The reason the patients are, the clots are difficult to actually dissolve, the reason these amyloids are being tagged by ubiquitinization proteins, but they are not being cleared by proteasome or endosome mechanism. Joshim, answer to your question is, it's such a new thing, you don't have an enzyme for that. You take 1,000 years or more than that to evolve to form an enzyme for it.
But one more comment to that. We are like we've been discussing that almost for a year now, that we're looking at multi-component fibers, and that is now the proof for that. So if you have a multi-component fiber consisting of a spike protein of amyloid plaque or amyloid fibers or beta and neutrophil elastase, then taking away the building material of that, like stopping spike from spreading, calming down the neutrophils and bringing down the elastase, and also reducing amyloid beta, which we can do with the molecules. So we do have a chance of cutting that cycle, at least prevent it from forming.
Very right. And you know what those compounds that you saw in chemical structures do? They actually remove the amyloid fibrils. Okay, so tell me where the spike protein will dissolve with. When it comes to prevention of formation, yes, it is very much possible. But I'm alerting my listeners. Okay, once formed those superfibrils, you can't dissolve it. It's very hard because we don't know what it is. So if you treat them with the chemicals that I showed you on different slides, the amyloid fibril would clear, it would go bind the spike protein. So you don't allow the ingredients to mix and make that superfibril thing. That's, Josham, very much possible.
Yeah, but one more comment to that. We had the same, where we couldn't observe it better than in the brain is, of course, in the blood. And so we had these results that were done on a patient where you could see that a triple anticoagulant would not do much to these thrombi because of that reason. Because they don't work on these mechanisms. And then we had them take some nutraceuticals only for 3 weeks, and they would have a insignificant reduction in these blood clottings. So I think there is hope, and we can measure it actually in the blood, not in the brain, because that's so difficult.
Very difficult, because you cannot take a biopsy from a living guy, So I mean, dissolving them could be an issue, but first thing is that do not allow them to get formed in the first place.
Yeah. Okay, but that we can cover, and that looks pretty good.
Yeah, why not?